feeding in Darwin's finches to citrate metabolism in Lenski's long-term evolution experiment. I mean, this term is hard to define, but you know it when you see it. I'm going to side with the Supreme Court on this one. And my lab studies the recent evolution of novelty within a sympatric radiation of pupfishes endemic to a single island in the Bahamas. So it's only 20 kilometers long. They're endemic to these salt lakes in the center of the island. But I discovered that this island contains a scale-eating specialist pupfish. So 50% of their diet is the scales of other generalist pupfishes in the wild. They do this all day long, biting scales off of other specialists. Here you can see the scale eater. This is an unsuspecting generalist here. And I can't resist showing this to you in the lab too, feeding on a euthanized generalist. But look at the spray of scales coming off the side of that fish. So this is novelty. So scale eating has evolved in other fish groups out there, but among the thousands of cyprinodontiform and aphrodinomorphs across the planet, this is the only known scale eating specialist. Now to put that in perspective, I propose the Ecological Novelty Index as a quantitative measure of novelty. But this is just the evolutionary distance from scale-eating pupfish to the most closely related specialized scale eater, which we find in a clade of Tanganyikan cichlids, but that's 68 million years away. And relative to the vampire finch, it's only about 20 million years away to other blood-feeding birds like this oxpecker. Now there's another specialist in the radiation, a molluscivore, and here you can see the differences in craniomorphology among these three species, as well as this generalist, which is widespread across the Caribbean. So massive jaws in the scale eater, and this novel nasal protrusion in the molluscivore specialist. So here he is in the lab. I think that this nasal protrusion actually functions as a stabilizing wedge, helping them to break open snails as they shake their heads from side to side. And to support this, there's actually another molluscivore out there with a novel nasal protrusion. And again, the distance to a similar nasal protrusion is actually the same, and it's another Tanganyikan endemic cichlid. So I hope I've convinced you these are examples of novelty. And sure enough, when we look at trait diversification rates shifting across the pupfish tree of life, we see that lineages leading to the scale-eating specialists are diversifying in oral jaw size more than 1,000 times faster than rates on neighboring islands. And here I'm showing my morphogram. Across these traits that I've measured, we see that trait diversification rates, the highest, are concentrated in the oral jaws. So there's only one other place across the entire range of cyprinodon pupfishes where we see sympatric species flocks, and that's in the center of the Yucatan. But this begs this question of why is trophic novelty so rare in cyprinodon pupfishes? That's sort of the central overarching question of my lab right now. So for this talk, I'll actually go over 4 different possible constraints on the evolution of novelty. And I also want to take this time to reflect on the value of being wrong. So, courtesy of hyperbole and a half, this is the wrong way to care for your pet fish. And 3 out of our 4 beloved theories here make predictions that don't hold up in this system. So throughout this talk, I want you to ask yourself, is that because we're sort of capturing novelty in action here? Is that why these processes are so different? Or maybe that most of what we know about these theories is from macroevolutionary pattern, whereas in this case, this is a recent evolution of novelty. So first, our beloved theory, like our beloved pet fish, first one is that increased ecological opportunity should drive adaptive radiation. So is that what we see? Is ecological opportunity unique to San Salvador in these salt lakes? To test this, I went to all of these neighboring Bahamian islands across the Caribbean as well as Dominican Republic. And across all of these habitats, here's salt lakes on San Salvador supporting trophic specialists. And here on neighboring islands, they look identical. They actually have identical competing fish communities. They're ringed in mangroves, they're all carbonate geology, they're all salt lakes. And everything I look at, there's similar lake areas, there's similar dietary isotopic diversity, similar stomach content diversity, similar trophic morphology. Especially consider this, that in brown here is this huge range across the Caribbean of trophic morphology for 30 craniofacial traits. This is a discriminant morphospace, almost exactly overlapping with the generalists on San Salvador. This really is just one wide-ranging species. Then on San Salvador, we get these two novel specialists, the molluscivore and the scale-eating specialist. The only difference I've found so far is subtle, a couple extra rare macroalgae species in some of the lakes on San Salvador, and anecdotally, there's thick mats of macroalgae in some places. So suggesting a possible link between increased ecosystem productivity and some trigger of these exceptional trait diversification rates. But that would be really weird, and especially this other radiation only has a single macroalgae species in it. So maybe not ecological opportunity. What about ecological release? So this is another beloved theory that in a new environment there should be relaxed selection on these generalists, enabling them to colonize new niches. So is that what we see? So from my previous talks, you've heard that I was crazy enough to actually breed 2,000 F2 hybrids in the lab, take these back to San Salvador Island, re-release them to the same lakes, to these field enclosures, track their survival and growth in the wild. And this is sort of what that felt like. This is a summary of the experiment. And they did survive. So here's the high-density fitness enclosure for one lake. So I'm showing you discriminant morphospace with these three ellipses are the parental species for reference in 2 dimensions and 3 dimensions. And then these little dots here in gray are all the F2 hybrids that died, as well as the survivors in black. But then fitting this thin plate spline to these survival data, we see that this fitness landscape is extremely complex. So molluscivores on the highest fitness peak, separated by a fitness valley to this generalist, which actually sits on this isolated fitness peak. And scale eaters are down here. They do terrible in all enclosures and all experiments. I'm not quite sure why. But I want to point out that instead of relaxed selection on these generalists, we see a high density of generalist type phenotypes, but these are doing very well. So no stabilizing selection on generalists despite their high frequency, even though we expect to see disruptive selection. So why is that? So the predictions of negative frequency dependent disruptive selection, and I just want to go back to this plot here, is actually that all of these individuals at the edges of the morphospace here should be surviving really, really well. That's the opposite of what we see from this experiment. And I have sort of a guess now, is that when we start to look at, I wanted to look at not just the effects of phenotype, but also the density of similar phenotypes within these enclosures. So a metric of that is just the distance to your nearest phenotypic neighbors. So that's what this x-axis is. It's just sort of density of similar hybrid phenotypes within these enclosures. What we expect to see is just a linear increase. So the fewer competitors you have in enclosures, the better you should do. Instead, we see a nonlinear rise. So that's true over short phenotypic scales. But as these hybrids get weirder and weirder, as they get to the edges of that morphospace, they start to do horrible in these enclosures. And that's replicated across both lake replicates. And this is just a visualization, but using general additive modeling, incorporating just phenotype, or a spline term for frequency, or a linear term for frequency, there's marginal support that the spline fit is actually the best explanation for this. So in summary, and I love to just show this figure too, to remind you that I measured 16 different phenotypic traits on these hybrids. This is actually a 16-dimensional space. And what these are is essentially cutting slices through that space and looking at the survival fitness landscape for that first enclosure I showed you, which was just a summary. But we actually look at 2-dimensional sort of slices through there. We see these fitness landscapes are very different, right? It's a fitness hyperdimensional volume. But they range from flat to up to 3 different fitness peaks. So in summary, instead of ecological release, we see stabilizing selection on generalists. This could be due to negative frequency-dependent disruptive selection having only a local scale and only on some phenotypes for some functional traits, right? Some slices through that space. So the next part, what about genetic diversity? So after it was an ecological opportunity, I would have sworn that populations with increased genetic diversity are more likely to radiate to cross fitness valleys. That's what we expect to see. And if we look at San Salvador Island, we see all of these salt lakes across the island. I was really expecting massive effective population sizes on San Salvador and nowhere else. And looking at genetic diversity across these populations, that's not at all what we see. So here are the neighboring islands in brown, and their sort of estimates of genetic diversity are all across the board, just like the populations in San Salvador. And in retrospect, this now makes sense, because essentially gene flow is everywhere. It's widespread across the Caribbean. There's lots of evidence for this using F4 statistics of gene flow in San Salvador, essentially homogenizing genetic diversity across this Caribbean-wide population of generalists. And even this species tree model is insufficient to really depict these relationships. So the better model is actually tree mix population graphs, where fitting up to 20 admixture events seems to be the best supported models for describing gene flow across the Caribbean. So this is sort of what I want you to be thinking of in terms of San Salvador is nested within these outgroup populations, and there's lots of gene flow, probably ongoing, not point events. So genetic diversity genome-wide is not limiting adaptive radiation or novelty, but it still could be limitation of specific adaptive alleles that are not on San Salvador but found elsewhere. So now I want to advertise my graduate student Joe McGurr's talk later today at 2:30. Joe is actually looking at the genetic basis of these novel trophic specialists, right? What does it take to make a trophic specialist? And out of 12 million SNPs, Joe has found across 41 genomes, Joe has found it takes very few differentiated regions, only in the hundreds, to go from generalist to scalating and molecular phenotypes. And he's had remarkable success at basically having high resolution of these regions. So this is just our classic FST differentiation, but there's only 2 fixed SNPs here in one of the specialists, centered within a gene that's been linked to craniofacial development. And we see increased DXY in this region. We see evidence of a hard selective sweep. So this is a really strong candidate, we think, for selection on these specialists. So finally, genetic architecture. So effect size, source of these potentially adaptive alleles within the radiation. Now here our theory is that large effect staining genetic variation is best for crossing fitness valleys. Is that what we see? So we've started to do preliminary QTL mapping in this system. So this is an F2 intercross between the 2 trophic specialists. But here I'm showing you the morphogram for 8 of the traits that actually had significant QTL, even though this is a small sample size. But we actually had power to detect these because they have moderate effects on explaining these traits. So up to 15% of the variation in jaw length is explained by a single QTL. And when we look across the morphogram here, we see again that the largest effect sizes are concentrated in the oral jaws. And just for comparison here, you may remember this previous plot, but I think it is interesting that there's a parallel here between effect size of the genetic architecture and these macroevolutionary diversification rates. And we don't know the direction of causality, but it's still intriguing. So finally, we have some support of moderate effect loci being important in this radiation. What about the source? Is it standing or de novo? And here I want to advertise my grad student Emily Richards' talk in this very same session later today at 3:30. Emily has found evidence that adaptive introgression from distant Caribbean islands actually was important. And she's also shown that this, again, is wrong. This is a construct genome-wide, but of course different regions of the genome have different histories. And some of these histories correspond to just the scale leaders separated from everything else in the Caribbean. I'll cut it off there. Thank you all.